So I am going to thank you for the nice introduction. I'm going to talk to you today about something that we've been working on the last couple of years, since COVID pretty much, about sort of a project that's come together because of a need that was pressing for the university and for pretty much all of society. We needed to be able to test COVID samples. And so one of the reasons that I'm heavily involved in this is because I just happen, by happenstance, to have a lot of the expertise needed to bring something like this together. I became excited about research when I was an undergraduate student. And much like all of you here, I did a number of summer programs. I stayed in a research lab at my institution in New York. And I was in one lab over the course of a couple years. And I applied, at the time, I didn't know that much about research as far as like what it took to become a professor. I had a mentor, but I knew a lot more about like the path for medicine. So at the time I applied to medical school, thinking that I might do a year of research. And that's pretty much exactly what I did. I applied to medical school. I ended up at UCLA and I took a year off in the middle of medical school to pursue a year of research. There's a lot of these one-year fellowship programs. And it was like that year where I really decided I wanted to do research more than I really wanted to do clinical medicine. And I ended up applying and joining our MD PhD program, our MSTP program. And I've pretty much been here at UCLA doing a combination of clinical and research since then. When I finished, I ended up as a resident here at in pathology, and that's where my computational interests kind of lined up. I did a lot of work with our informatics program, and ultimately started my lab after I finished my training. So I do about 10% of clinical time right now, and I do the vast majority of my time is spent in more research and translational endeavors. My lab, when I started it, was around genetics of rare disorders. So if you know any of the students who are working on projects in my lab, you'll notice that they're all working on these sort of exceedingly rare disorders. But what I'm going to talk to you today is sort of about a project that came together from 3 different departments, sort of people with different expertises, and how a lot of things kind of came together at the right time with the right set of people to put together the testing program that we've been running for it's been about 8 months now, it's been almost a year, and sort of what that takes from both a research and development side, but also from a clinical standpoint. And I'm happy to answer any questions if people want to email me. I'm happy to set up time to chat if people want to talk about career paths. But so when you think about diagnostic testing, all of you are aware of COVID tests, because I'm sure every single person who's listening has had at least one. And when we do testing, people talk a lot about how fast you get your results back, how sensitive it is, if it's a PCR test. But there's really 3 things we think about in the world of testing. We think about how rapid your result is. Do you get a result within a minute or 10 minutes, kind of like a pregnancy test? Do you get a result that's very sensitive? So how little virus can you have in that tube that we can detect? How much do you need to be detectable? And then how scalable it is. If you want to test one person, it's always easy to do a test on one person or one sample. But if you want to test thousands of people, that's very different. And if you want to test hundreds of thousands of people, that's also on a different sort of scale. So you can think of it like, I want to bake 1 loaf of bread. That's fairly easy. It might take you the same amount of time as baking 3 loaves of bread if you did it in parallel. But what if you wanted to bake 3,000 loaves of bread? You'd probably want a very different system than what you're doing for doing 1 loaf of bread. So there's the trade-offs. No test does all 3. So all of the testing that we do right now, it's usually 2 of the 3 or 1 of the 3, but if you find something that's both rapid, sensitive, and scalable, you've hit the jackpot, and you should start a company and monetize that, because that's sort of like the holy grail of testing. So we are really thinking about SwabSeq because we developed it to be scalable and sensitive. Its turnaround time is rapid, but I would argue that if you're really looking for a rapid test, there are some pretty good tests out there that are as rapid, if not faster, than the turnaround time for SwabSeq. So this assay was really born out of collaborations that had started many, many years ago. And Sri Kasuri is a faculty member in chemistry and biochemistry. And he's recently left about a year ago to start a startup company called Octant Bio, which really uses scalable sequencing to identify drug targets. And so during their during this time when COVID had started up, all of San Francisco was closed, everybody was working from home, and they thought that, hey, our assay would work as a diagnostic platform. And so they started doing some small pilot experiments, and what you can see here is this is like I think I took this from his Twitter account, but they were pretty much looking for qPCR machines so that they could test out whether or not their protocol would work if you changed it so that you tested for COVID rather than some G protein coupled receptor. So short story, they changed the primers. It's very transferable and translatable, and so they were able to sort of show that proof of concept that this could work if we were to tweak it a little bit more. And so this is SwabSeq. SwabSeq, this is the method we use right now for testing at UCLA. I know most of you are not on campus, but what happens is we obtain a specimen, so a specimen like saliva or swabs in a tube. Each specimen goes into one plate of a 384-well PCR plate, and that gets run on a thermocycler. So this is where we take that viral RNA and we try to make lots of copies of it so we can see it. But at the same time that we're making all these copies, we're adding a molecular barcode, so a genetic barcode, so that we can identify exactly which well that sample came from. We then multiplex the samples, so we pretty much pool the samples together. We dump everything into one tube. So you go from having a plate, and because we've barcoded each sample, now you have one tube with up 384, up to 100,000 samples in theory. And then you take the sample and you run it on a sequencer. Once we sequence the sample, you then take those barcodes and you can deconvolute. You can figure out which well that read came from, and then we can do our testing. So we can just tell you if there is SARS-CoV-2 virus reads there, or if we just detect the control reads. So one of the major differences between this and what you typically think of in traditional RT-qPCR testing is we don't have to do an RNA extraction. So most of the protocols out there, if you were following a lot of the work out of the CDC early on, we were really limited by RNA extraction reagents. So we had labs emailing the research labs saying, do you guys have extra reagents for RNA extraction because we're running out? So those were really in really short supply early on, and it adds extra time to the workflow. We do genomic barcoding of samples. So when you think about what happens in a typical clinical lab, they add a fluorescence fluorophore to the sample, and then if you see a lot of SARS-CoV-2, that fluorophore will start to light up, and that's what you detect. The last thing is we do an endpoint readout of PCR. So we put less primer and we use up all the primer, and that allows us to normalize the sample early on. And that's going a little bit into the weeds of the technical details, but it's important because as we move forward, if you've worked in genomic technology, the normalization step can be a real pain in the butt, and so we do a lot of things to make the process as easy as possible for testing. So this is what our amplicon looks like. You have your target sequence. So this for us is SARS-CoV-2. We also have a control that we use to make sure that we got a human sample in there. And this uses we have a PCR primer, but the barcodes are attached to that primer. So you have I7 and I5 barcodes, and then we have these adapters which allow us to sequence it on a Illumina sequencer. After we sequence, like I said, you can actually see which samples have a large number of coronavirus reads, and if you've passed a certain threshold, we call your sample positive, and that's how we decide whether or not you get a positive or a negative result. On our end, when we look at the data, we can actually see quantitation of how much sample you have or how much virus probably was in the sample originally, but we don't report that out because it's sort of hard. There's no clinical reason to do that. And also, we use unique dual barcodes. So any of you who are thinking about combinatorial indexing, there's a lot of controversy around whether or not you should do semi-combinatorial barcodes so you could expand the ways that you combine your barcodes on each end. But we chose to do unique dual indices because there's a little bit of an idiosyncrasy with the Illumina sequencer. They have this thing called barcode hopping, where the barcodes can actually swap between samples, and then you could get a false positive. So we don't like false positives or false negatives. We want to be as accurate as possible, and so we use unique dual indices. And then one of the things that makes this assay unique, and one of the reasons we don't use sequencing a lot for quantitative assays, is because there's a lot of variability between samples. So if you think about my sample versus any of your samples, my saliva has a certain set of inhibitors based on what I ate, what my genetics are, and probably a lot of other things. And it's different from yours. And so if we were to put our saliva into the same set of reagents to try and amplify something, it would amplify at different levels because of the inhibitors to the polymerase process, the polymerase and just the PCR process. So in order to quantify, to do a real quantitation, we have to use these in vitro RNA standards. So what we did is we took that region of the S virus that we sequence, and we created a synthetic version of that RNA. So we have a version of it where we take 6 base pairs here in red, and we actually flip it so that you can read this ATGCGT, and now it says, we flipped it, so now this goes here, and it's TGCGTA. So this synthesized S sequence, this quote-unquote spike in, allows us to normalize it. If you have no SARS-CoV-2 in the sample that you provided, then we'll still sequence something, we'll still amplify and sequence something. So this allows us to know that the primer was there, the primer works. And it also allows us to then normalize any SARS-CoV-2 sequence that we see. So essentially these two things will compete against each other within that reaction, and allows us to call a negative sample in the well. So this is, you can kind of see what it's looking at here. We're going to look at a lot of these graphs. So on the bottom, it's the copies of virus per reaction. So we created these synthetic versions of this of each sample to see how accurate our assay is. And then on the y-axis for this graph, the y-axis for this graph, you can see this is just the number of SARS-CoV-2 reads that you see. So you can see there's a pretty wide spread, right? This is on a log scale, but you can see that it's a pretty large spread between what should be 4 copies of virus per reaction. Now, if you normalize it, so if you normalize it to this standard spike that's present at the same level of every well, you can see that it sort of tightens up a little bit. So our ratio is actually probably better because whatever's happening to the spike is happening to the virus sequence as well. And so we like this a lot because if you count reads, it's just noisy. It's subject to different amounts of PCR inhibition. And that's one of the reasons why we don't usually quantify things by sequencing counts alone between samples. But if you normalize to this internal control, it actually helps turn this reaction from something that is just a sequencing read reaction to something that's ratiometric and does exactly what you see here. It tightens up that range of the ratio. So just to summarize everything I've tried to explain about this assay, we have 2 controls. We have a synthetic spike, which has that flipped 6 base pairs. So this allows us to make sure that our S2 virus PCR is working. And also we have this human RPP control. This tells us that there was human sample that was put into that well, and it wasn't a sample from another animal, or it wasn't just an empty, like we didn't get any sample for whatever reason. And then we do all these, we do a ratiometric readout, we do endpoint PCR for normalization at this sequencing level. So we're allowed to call true negatives. We know that every sample gets a lot of reads, and we just have fewer artifacts. So it's a little bit more robust in that sense. Okay, so the next couple of slides are going to show you how this assay now performs compared to the traditional qPCR that they do at the UCLA Clinical Microbiology Lab. And so this is a really important thing to do when you're developing a test or a diagnostic. You want to know exactly how your test performs to what's already established out there. So for us, it was the nasal swab, so the nasopharyngeal swab, so the swab that goes all the way to the back of the nose, practically brushing your brain, that's purified in the lab and then sequenced. So we did our assay to look at what our limit of detection is when we were looking at pure RNA purified samples. And what you see here is that in A, we went up to copies of virus per mil, up to 8,000 copies per mil. You can see that our ratio holds pretty nicely, but that we have a nice limit of detection around 250 copies per mil of liquid. So per milliliter. So that's a pretty small, it's like maybe the size of my thumb, that much liquid, and we can detect down to 250 virus copies there. That's pretty good because around that's where the limit of detection for the Stanford and Berkeley assays are as well. And then we did the same thing, except we took samples from the UCLA clinical lab that they had already processed. They gave us positive samples, they gave us negative samples, and we ran both of them, and using our limit of detection, we were able to call all the negatives negative and all the positives positive. So this is really promising from this point of view, but we really wanted to know if it works in extraction-free protocols, because every single step that you add to a clinical protocol requires validation, it's extra work, and we know that the sequencing process takes 5 to 6 hours. So we were spending a lot of time on the sequencing, and we wanted to be able to skip the extraction step, which is very labor intensive and supply chain limited. So the first thing we tried was, can we just take the nasal swab and not RNA extract the liquid and stick it directly into the sample? So what you can see here, the short answer is yes. When we do this, our limit of detection is about 558 copies per mil. So this is a nasal swab that goes into a liquid called tris EDTA, which is a standard buffer in the research space. It's not actually a standard liquid in the clinical lab space, which we'll talk about a little bit as to why we don't use this commonly. And then the other thing that we did was we took those same samples that we collected, we tested them. We took our samples, we collected them from patients who we knew to be positive, and then we sent them to the clinical lab for extraction, and then we also ran them by our assay without any extraction. And so what you can see here is we call the positives positive and the negatives negative. So our test works in extraction-free swabs. And then there was a lot of people were upset because nobody wants to stick a swab up their nose. I don't care how comfortable the swab is. And so and at the time when we were developing this, there was a lot of interest around saliva direct. And so what we opted to do was an extraction-free protocol because they're cheap. Everyone, in theory, can spit into a tube. And this is something that's easy enough for, I think I've heard people as young as 3 are able to spit into a saliva if you give them a little candy. And this is less affected by supply chains because we just collect the saliva neat. We don't add anything to it. There's nothing special. It's just saliva in a tube. It's just kind of gross. So what we do to inactivate the RNases and to make sure that we can detect the virus is we heat the saliva up to 95 degrees. And so you can see here when we have our contrived samples, our limit of detection is a little bit lower. It's about 1,000 copies per mil. And when we are able to compare the saliva that we do by swab-seq versus purified RNA from saliva, an MP swab that's done at the same time, you can see that we detect almost all of the positives. So there's a handful at the lower limit of detection that we're not able to detect. And we are able to call all of the negatives we call negative except for this one. So it's a pretty good test. It's not as sensitive, but it's a lot more scalable. So about a little more than a year ago now, we kind of knew that the technology works, and it's interesting to think that the technology was really fairly straightforward as far as diagnostics go, but what we came to realize was the logistics are sort of equally, if not more, challenging. So to do testing, so if you're in a research lab, you can sort of do whatever you want. I can take my spit, I can test it out, I can add a little bit of SARS-CoV-2 RNA and see if it can be detected. But once you start thinking about doing this test as a diagnostic, the game totally changes. So the game becomes one of those, you need to be able to do your test. If you're going to give people results that they're going to then act on clinically in a medical environment, you have to do your test in what they call a CLIA lab. So CLIA is a federal regulation that came out in 1988, and it's a set of rules that every lab has to adhere to. You get inspected, you have to have the certificate. It's a little bit of it's a pretty big deal space-wise and money-wise, and generally we don't mix the two. You don't mix clinical stuff with research stuff. You have dedicated areas for each. So to bring up a lab, you need space, you need money. There's a lot of paperwork involved. Every single thing that you do in the lab has to have a standard operating procedure. You have to validate every test, and you have to have your test done by certified personnel. So the people who are working in the lab have gone through a year of training and have passed the test and are licensed by the state in order to perform this testing. So there's a lot of these regulatory things that people don't necessarily think about when you're bringing up a test, because this is the part where things get very expensive, because all of these pieces have a premium and you essentially have to have 1 or 2 dedicated people to make sure that you're in compliance, because if the state comes in and does a surprise inspection, you could get shut down. So in order to process all of our samples, we wanted to bring up our clinical lab in a way that was what we like to think of as semi-automated. And I think given what we knew back then, we thought we would be testing about 6,000 samples a day. And we said, well, why don't we try to make most of it automated, but still have people in the lab, because it's hard to do a fully automated system. So we have right now, people will collect their sample into the same type of tube. This gets racked, and we pay a company to register all the samples, and people we pay a company to kind of handle the registration process. So we can link a tube with a sample to a barcode, and that barcode is linked to a person. We take these racks, and they come to us in racks. We put them into the water bath for an activation. These then go into an automated decapper and into our PCR mix. To collect our samples, we actually started off looking around to see how we could get samples into the tubes. I found these on Amazon. They are actually mini plastic funnels that were sort of advertised as for filling perfumes into little bottles or little art projects for kids, but turns out they fit perfectly into our tubes, and so we pretty much bought out all of Amazon's stock at some point in order to purchase these. A lot of other companies were selling us more fancy devices that were about $1 or $2 a piece, and these are only 15 cents a piece. Then the sample comes into our lab, and they're registered at the site. They come into our lab with 2 barcodes. There's a linear barcode and a 2D barcode here at the bottom. It registers through this software called Precise MDX. I think they use it at a lot of universities at this point. And then our lab then scans it in. If you have a nasal swab, the swab is actually collected inside the tube as well, and we can pipette around it. And the other thing that we did as part of this is as part of optimizing all the processes, it was impossible to get nasal swabs early on, and all the nasal swabs were built for tubes that were about 5 times the size of the tube that we wanted to use. So we designed a special swab that had a breakpoint pretty close to the swab tip. So you put this part into your nose, you break it off here, and you deposit that into the tube. And this was designed by one of the surgical residents who was helping us. And we ended up having it 3D printed because we really wanted our process to be simple, and we didn't want to have to be handling a lot of infectious sample because it's just dangerous. It's COVID. So our process is really divided into 2 steps, and I think the easiest way for you to kind of see what these 2 steps are, we do pre-PCR. This is when we're handling the tubes and the samples and trying to get it so that we can amplify it. And then we have post-PCR, which is where we have tons of PCR products everywhere, and this process has to be done in a completely separate room because it can cross-contaminate into our pre-PCR process and just cause us to have a lot of artifact in our sample. So we have a separate room for the post-PCR. So I'm going to show you, hopefully this YouTube video will work. We have a little video. Can you hear it?
First, we heat the samples at 95 degrees for 30 minutes. We use a flatbed scanner to record the location and barcode number of each tube. Then, we use an automated decapper to remove the caps from 96 tubes at once. Using the 96 well pipetter, we add sample to each well of a 384 well plate that has been prepared with primers and master mix. Finally, we load and start the thermal cycler.
So this process from start to finish is pretty fast from the time we start processing until we finish our PCR step. Let me get to so the next thing we do is, once we have all those plates out, we then do the post-PCR. So we do this process, we do several sequencing runs a day, and this process is simply to take all the samples and mix them all together.
First, we remove the plates from the thermal cycler. Using the Integra vial flow, we pour all of our barcoded samples together into a single reservoir. Next, we transfer the pooled PCR product into a conical tube and vortex. Then we use magnetic beads to purify our library. We use 2 bead purification steps to isolate fragments between 100 and 200 base pairs. Next, we measure the concentration of the library using the qubit and adjust it to the appropriate concentration for the sequencer. Finally, we load the library and the sequencing primers into the sequencing cartridge and start the sequencing.
So that's the whole process. I mean, we shortened it for the sake of time, but from start to finish, it takes about 12 hours once it gets into the lab for us to result the sample. And in order to accommodate the need that we thought we would be handling, and which we appear to still be handling, we purchased upfront 1 million funnels and created thousands of test kits. This is what 200,000 tubes look like. Getting our supply chain finalized was probably one of the more challenging parts of this. So we started with this process back in April 2020 and validated the technology. We had an FDA EUA by October of last year, and we actually started testing for UC Santa Barbara in November. We sort of did their overflow testing. So we were testing right as the surge in the winter started, and we tested from December, and we're still testing now. Our volume had dropped over the course of time because a lot of places were doing less testing, but now we're testing, we're starting to pick up testing again with the surges we're seeing in the Delta variant. So how we've gotten our samples has changed a lot. Initially, people were collecting at specified sites. At this point, we have a vending machine version of this testing where people are getting their kits from the vending machine, self-collecting, self-registering, and then dropping it back off in this little, not a trash can, it's like a little bin for all the testing tubes. So this is being done all across campus for anyone who's here every week on main campus, across the camps, and across, we're also testing for LAUSD, which has just started. So this is what our kit looks like. We've tried to simplify it. As you can see, we're not doing the nasal swab anymore because we actually had a hard time getting people to break off the swab at the right spot, which made testing pretty challenging. So right now we're only doing saliva testing, which is pretty equivalent to this nasal swab. And for us, the processing is a lot easier on that end. So a lot of people have asked me over the course, and this is going to be sort of the last thing I discuss. If you are vaccinated, what happens to your infection rate? Does it help? Do you have less virus if you are infected? So we did look at a set of data from UCLA Health. So we started vaccinating UCLA Health employees in mid-December as soon as the vaccine came out. That was in the midst of the surge when we were testing people weekly. We looked to see when people got vaccinated with their first and second dose, and then we looked to see what the positivity rate was. So if you look at our positivity rate, you can see that it was highest in that first couple weeks of right at the new year, mid-December all the way through and end of January was where we really peaked in that winter surge. And then it really died down at UCLA Health. And what we found was if we did testing, so people were tested by our clinical lab, they were tested by SwabSeq. So you can see a little bit, there's some differences, and we just converted our ratio into what we call a cycle threshold. So the way to think about the cycle threshold is the higher it is, the less amount of virus you have. And then along the x-axis, we're looking at weeks away from first and second dose. So this is when the first dose, people who a week out from their first dose, what their viral load was, and so on. And then the same thing for second dose. So you should notice two things on this graph. First is that after the first dose, you can see there starts to be a trend where the CT values are a little bit higher, meaning that they have a little bit less virus that we're detecting. And this is on a log scale, log 2 scale. And then after the second dose, we see far fewer positives And we also see that trend sort of is maintained. So what we saw, and this is all from several like months ago now, before the Delta variant, we saw that it was very protective of infection and also against sort of increasing, slightly increased virus CT scan or lower viral load. So we see this is not actually what we see with Delta. With Delta, we actually see much higher viral loads, but we don't have the vaccination data to show whether or not it's changing, whether or not how the vaccine interacts with it. And there's a couple confounders. We don't know how long ago someone had a vaccine. So there's a lot of differences if we were to do the same type of study now. But the one thing that's very clear is that once you're vaccinated, even the first dose, there's some protection. The second dose is much better. You're much less likely to even test positive. And I think that's held true across all studies that have been done for COVID-19 across the country. So I'm going to skip this slide because I pretty much said, so if I know many of you are not here, but if you haven't been vaccinated, please get vaccinated. I'm just going to make that plug because I've seen how effective it is in our testing program. And it's really just an important thing to do for everyone around you. So if you have questions about the vaccine or about this test, I'm happy to answer them. You can ask them here, or you can always email me. My email is there. And then I just want to acknowledge all the people, because this was 100% like a huge collaboration across multiple departments within pathology, human genetics, computational med. And so these are all the folks that were kind of critical at some point across this entire project, and many of us are still working on it today. When we brought up the clinical lab, we hired a whole new team of lab professionals to really run the clinical lab. And so this is a lot of the people who have been working on this sort of for the past 6 or 7 months, day and night, trying to make sure our testing is going smoothly. And then if you want some more information, there's a number of preprints out. So they're all on, I think, medRxiv, and maybe one is on bioRxiv. So I'm happy to answer any questions. And I thought I saw one question